Can you hear me now? Okay, good. Sorry, I forgot the buttons. Still trying to get used to all the buttons that I have to push. Okay, sorry. So maybe let me start again. Actually, I intend to show you the movie, the video. Maybe I'll show it to you next time, next week. But just to tell you that there's a class of so-called the killer marine snails that can catch fish. They use their harpoon to inject the toxin into the fish, so that to paralyze them, acting on the nervous system, so that they can eat those fish. Now the scientist, Professor Oliveira, actually studying those drugs, those toxin in the snails, and then use that for development of drugs for pain. Actually, there's a very interesting story behind how he started that project and then start to develop some of the wonderful drugs. Actually, he is actually a visiting professor in HKUST, and actually he has given some talk before. If you are interested, you can take this link and then you can watch those video and his presentation. It's very, very, very interesting. Now, I bring out this story actually is to tell you that now we have started to talk about the neurotransmitter, and you will see that how some of the animals can hijack those systems to achieve what they want. For example, for toxin from the snake, toxins from the spider, and so on. Okay. Now the first, let me start. So today we are going to talk about acetylcholine. Acetylcholine is one of the most studied neurotransmitter, particularly in the peripheral. Now, but however, in the central nervous system, this neurotransmitter is less understood actually, but the mechanism and the functions. We will talk about a lot of those different neurotransmitter. Now, a lot of those peripheral actually is much easier to study because that involves like physiological functions. Okay, for example, in the parasympathetic system or sympathetic system, or in the muscle system. However, in the central nervous system, that involves your cognitive functions, then those will be more difficult to study. For example, which neurotransmitter is related about memory formations. Okay, now that will involve a much more difficult task to investigate. But anyway, we will start with this the most well-studied neurotransmitter, acetylcholine. Now it is also the first neurotransmitter that ever to be identified. Now because it controls your skeletal muscle contractions. Okay, it's a small molecule neurotransmitter, but with a wide variety of functions. Okay, so you'll see that actually control a lot of different things. Now, there are two major classes of receptors, which is nicotinic, and then the other one is muscarinic. So, the nicotine and muscarine, those are the compounds. Now, in the early days, so how can you label those receptors? So you use some of the compound extracted from plant. Okay. Now, plant, in the other hand, they also produce a lot of those chemicals that binds to those receptor. Now, one of the reasons behind is actually that may provide protection for them, so that some of the insects or other things that will not eat them. Okay, so that's one of the possibility. Now, but when we study the receptors, if you still remember, when we talk about the synapse, so you have to get something that can bind to it, is it? Okay. So that's the reason why that when the very beginning, scientists find that nicotine and muscarine can bind to two classes of receptors that all can bind to acetylcholine. Is it okay? So when you study the receptor, there's always this kind of the case. So those receptors that binds to nicotine is called the nicotinic receptors. Those can bind to muscarine, is muscarinic receptors. Now, they both can be activated by acetylcholine, okay? However, their structure will also be recognized by the nicotine and muscarine, okay? Those are also their agonists as well, okay? Now, in the classes of these two classes of receptors, they have different distributions. For example, all skeletal muscle use the. Oh, sorry. Yes. Today is strange. I forgot a lot of different things. So, sorry. So now it's not the online student, it's the face-to-face student. Is it okay? Okay. So the nicotinic receptors actually, so next time just let me know, because sometimes I think. No, okay. So the nicotinic receptors, actually distributions, all the skeletal muscle, okay, and the striatal muscle as well, some of those in the head, facial muscle and chewing. If you still remember when we talk about the parasympathetic and sympathetic system, then some of the students ask about, why there's a NIC, that's a nicotinic, why there's some is MUS, that's the muscarine. So those two classes of, receptors. So those are in the postganglionic, postganglionic parasympathetic system and preganglionic sympathetic system. So I hope that you still remember what is postganglionic and what is preganglionic. Still remember? Postganglionic is the one that, after the one that from the spinal cord. Preganglionic is the one that in the spinal cord going out, okay? So that's the reason why that the autonomic nervous system is still origin from the CNS, okay? Just in case that you kind of still a little bit confused, okay? Now, some cholinergic receptors in the cortex and subcortical structures, okay? Sacropostganglionic parasympathetic fibers, okay? Those have what is nicotinic receptors. And the muscarinic receptors, okay? Some of the parasympathetic innervated, smooth muscle, for example, some of the salivary gland and tear glands, okay? Now, in the central nervous system, there are also cholinergic receptors that are muscarinic. So you see that in the central nervous system, actually both the nicotinic and muscarinic are contained in receptors in the central nervous system. And other parasympathetic system, okay, for example, the bladder and the colon, for example, okay? So remember, both can be activated by acetylcholine. Now, but they can be classified by their binding of the other ligand, okay? Now, so this is a very interesting effect. That means that they are structurally different. However, they both can activated by acetylcholine, okay? And you can distinguish them by different ligands, okay? Now, this is the idea about those, the receptors and the agonists of the ligands, the agonists or antagonists. Is it okay? So ligands means that they can bind to the receptor, either in the same site or different site as the neurotransmitters, okay? So you'll get more and more of those in the class. Now, in the central nervous system, so here is the case. There are different projections from the different nuclei. So this is the septum, substantial nigra, corpus striatum, thalamus. Those are the bases. So you can see that the substantial nigra have some of the projection going to the thalamus. And there's a nucleus basilis that project all to the whole cortical region. Okay. There also in the septum, there's a group of acetylcholine neurons project to hippocampus. Okay. So those are the things that we know. Okay. However, the function that has been implicated in the formation of memory as well. Okay. So those are the places that are going into different. So if you still remember when we talk about, how to define neurotransmitter, there must be a place or nuclei, so such as those, that if you lesions that, then that will remove the neurotransmitters. Okay, that's the reason why that the case, if you still remember. Now, for every neurotransmitter, I will talk about, their synthesis, their storage, their release, and their degradations. Okay. Now, if you still remember in the neurotransmitter sections before we talked, we always talk about how we can modulate the transmissions of certain neurotransmissions for different level: the synthesis, the storage, the release, and the degradation. Okay. So for acetylcholine, let's look at it step by step. So everyone okay so far? Now for the synthesis, acetylcholine actually, if you look at it, is the choline and then also with acetyl group. So the synthesis requires the choline, which is uptaken enters neuron via the carrier-mediated transport. So that's the reason that choline actually is a very important component that we absorb in the food. Okay. The acetylation of the choline will require the acetyl coenzyme A. So if you still remember, coenzyme A always carry the acetyl group in the metabolism of the fatty acid, or in the metabolism of glucose. So this is by the enzyme called choline acetyltransferase. Now, if you still remember, when we talk about how classifications of the neurons can use the neurotransmitters. Now, this is the case in which that all those acetylcholinergic neurons will contain this enzyme. Okay, because they need this enzyme to synthesize the neurotransmitters. Okay. So each is cytosolic enzyme found only in cholinergic neurons. Okay. Now another term is cholinergic. Now cholinergic means that the neurons that use the acetylcholine as their neurotransmitters. Okay. So it's the acetyl coenzyme A is synthesized the primary in the mitochondria, and choline is supplied by a high affinity transport. So you can see that the choline actually will go up by the transporter, okay, active transporter going in, and then the mitochondria will provide the acetyl coenzyme A, so that the enzyme, the choline acetyltransferase, will then form the acetylcholine. Okay, so this is the biochemical synthesis. Now, how about the storage? Now, the storage will require the transporter that transport the acetylcholine synthesized in the cytosol into the vesicle. Okay, so here is a simplified diagram, and you have learned that in the synaptic transmissions, those vesicles will be uptaken and then go into the reservoir, and then that will go into the active zone and ready to release. Okay. Now you can see that this is the enzyme actually can go both ways. So in the vesicle, the acetylcholine will be protected not degraded, and also concentrated, and then ready for release. Okay. So after release, the acetylcholine is released by the calcium-mediated exocytosis. So those we discussed about. And then neuromuscular junctions, usually one nerve impulse will release around 100 to 500 vesicles. Those are in the quantum safe, same amount to be released. Now, once in the neuromuscular junctions, then they will bind to the receptor, so nicotinic acetylcholine receptor, that will open the ion channels, okay? And the channels will depolarize, and then that will become the end plate potential, and then that will make the muscle contract, okay? So that's the release, all right? So you can see that in this case, this is the presynaptic, preganglionic neurons, which is cholinergic, and then release that to the innervated to the postganglionic neurons. This is the sympathetic system, and you can see that. So this is a sympathetic ganglion. This is postganglionic neurons, and that will release to the target organs. Now you can see that regulations of the acetylcholine release is controlled by one of the ways is controlled by the uptake of choline. So if you inhibit the uptake of choline, then the stimulation, even with stimulations, the release will be slowly decreased because the use up of the neurotransmitters. Okay. So one of the special things is that actually in the acetylcholine, the vesicle contain ATP, and then that will co-release it out, and the ATP will be hydrolyzed and then become adenosine and the phosphate group. Now the adenosine can bind to the synaptic receptors. This act as back control back the release and the control of the overall synaptic transmissions. Okay, so if you release a lot, then this receptor will be activated, and then the terminal nodes that actually the synapse is quite active, so that it can enhance the transmissions or enhance this synthesis of the acetylcholine. Okay. So the main mechanism for the pharmacological block. So where can you block? So you can block the choline uptake, okay, or inhibit the acetylcholine release. This one is the one that actually Botox is working, in which it prevents the release of the acetylcholine. So that's the reason that it can prevent the muscle from contractions, okay? Or you can block the postsynaptic receptors or ion channels. Okay, we will have some more example of how we can block those. There are different drugs that can act on those. Or if there's a persistent postsynaptic depolarization, okay, that can happen. For example, if you stop that by stop the metabolism of the acetylcholine, in which we will talk more about it. So you can see that actually. So from the synthesis to the transport and storage, and then to the release, and then the last step will be the degradations. Okay, now all the neurotransmitters include those steps, and then all those steps can be affected, or finally in the receptor level. We'll talk more about those modulation. Now after release, the acetylcholine is degraded by extracellular acetylcholine esterase. So it's here, acetylcholine esterase, in which that will break down to the choline and the acetate. We will talk quite a bit on about the enzyme, okay? Because this enzyme actually has been studied thoroughly for a long time, because that acting on this enzyme basically hyperpolarize the postsynaptic receptors all the time. Okay. Now the release and then the metabolism of the acetylcholine is very fast actually. It can be hydrolyzed within 1 millisecond by the enzyme. Okay, so that's reasonable, because that if you don't stop it, then you cannot respond to the next step. Okay, for example, if you're moving, so like what I'm doing now, the muscle is contract and relax, contract and relax, contract and relax. So each one involve, you know, the excitations from the terminal. Okay. Is it okay, the online student? And the face-to-face student, so far, so good. Okay, very straightforward. If I'm too fast, then you let me know, okay? The online student as well, okay? So now we talk about the acetylcholine esterase. Okay, there are two main forms of the choline esterase. Both enzymes belong to the family of the serine hydrolase. Now, you can see that actually it's very important for us to have the enzyme everywhere. So if you still remember, I asked about those, some of you asked about, you know, where are those acetylcholine esterase or choline esterase? I said even in the red blood cell actually. And then because that your muscle contraction after that, if it is not metabolized, it can diffuse into the blood. And then if circulated, if you don't have enzyme to break down, then that will be a problem. And another problem, some of those, if you have something you have eaten and then their form, their structure is similar to acetylcholine, then that will be another problem that, you know, you need an enzyme to metabolize it to prevent acting on, you know, the muscle or the parasympathetic or sympathetic system. So there are two types, you know, the one is acetylcholine esterase. Now this one is more specific for acetylcholine, and it is also much more rapid hydrolysis. Okay, there are many membrane-bound enzymes. We'll talk about those this enzyme more in later slide with more details. Okay. The other one, butyryl choline esterase. This one is less selective. Now, this is in the occurs in the plasma and many tissues. The reason behind, as I mentioned, now first of all, it can metabolize acetylcholine, and also it can metabolize some similar structures compounds as a, you know, protective functions. Okay. Now, just to remind you that I still haven't received all the student forming group for the project. At the moment, I just have around 30 students, 30 or 31. So, so if you haven't formed your group yet, please do it as soon as possible. Otherwise, I will form the group for you. Okay. Okay, let's continue. Now you can see that actually the comparison of those two enzymes. Now, that actually has been predicted by Dell. You still know how about Dell, you know, the one that had Dell hypothesis, you know, that only one neurotransmitter per neurons, okay? But that's not the case actually, okay. Now, actually, he is the one that actually very beginning, you know, study the nervous system. So he predicted that actually exists. In later time, there's some drugs actually can act on the choline esterase, physostigmine. This one is actually one of the drugs that we will introduce later. Now, in 1932 and 1940, and Dr. Stedman actually purified those enzymes and then being studied. So you can see that actually it's long time already. Because that this is the enzyme, yes?
What does the A stand for in the first line?
Which one?
The E on the first one.
Tells existence of E. What is the E name?
Oh, the enzyme, existence of the enzyme. Yes, sorry. So what he means is actually, because there are two of those, acetylcholine esterase and butyryl esterase. Okay. Yes, good, yes?
Does he predict the existence of the esterase?
Yes. Yes. Because what he was thinking is that if there's a receptor for that, so there must be something that can have to stop it. Okay, so it's so remember they at the beginning they talk about there's a chemical, there's a synapse. Okay, so if there's a neurotransmitter release and it binds to the receptor, then there must be something that stops the actions. Okay.
So in other words, there must be a degradation system.
Yes, removal system. Yes, yes. Okay, if you have sent me the email, yeah, I didn't reply to you, but if you send an email and it's not returning, then you should get it. I should get it, yes. And if you can send it, yeah, you can send it through the Canvas actually. Yes, yeah. Then that will get to my email as well. Okay. Okay. Maybe I will reply to those that I have received your email later today. Okay, so that you know that I got it. Okay. Let's go back. So the characteristic comparisons. Now there are multiple forms. Acetylcholine esterase have more forms, in which that we will talk about it more than the butyryl esterase. But both can degrade the acetylcholine. Now distribution is everywhere, but acetylcholine esterase is more tissue specific, but still they are everywhere. Now both of them are vulnerable to organophosphorus inhibitions. But they are different by their sensitivity. So if you heard about organophosphorus inhibitions, you know, some of you may heard about from the news in which the vegetable it is illegal. Some of the organophosphorus insecticide are illegal because those are very strong. It is very good at killing the insect that eat the vegetable, but it will also affect human if we consume it. So there was, you know, some of the cases before. That's the reason why that, you know, all the farms that supply vegetable to Hong Kong needs to be have license, because that, you know, some farmers, they use some of the illegal insecticide, which is this class organophosphorus insecticide that kill the insect. Okay, so we will talk more about this class of the chemicals later. Now they have strong homology because they both use the same catalytic mechanism, including the serine, histidine, and glutamate. Okay, we will also talk more about those structure. So some of the further comparison, you can see that the size, they are actually very similar, not too big. And then there are glycosylation residues, and, you know, their molecular weight, you know, around 80 kilodalton to some of the 70 kilodalton, some of cysteine, and so on. So those are some of the characteristics of those. Now, this is the general structure of the acetylcholine esterase. So there are 6 exons. Okay. Now, they can be several molecular form, and depend on how they are attached to the membrane and the structure. Okay, they have before the stop codon, there are 3 splicing alternatives. Exon 2, 3, and 4 encode the core molecule that contain the catalytic residues. Okay. Now continue from the exon 4, that will be a monomeric species. Now it can extend to exon 5, then that will give the signal for glycophospholipids that can, binds to the phospholipid, get the signal. So for the exon 6, then they can form the with a cysteine linkage, they can form a link to different subunits. Okay, so this is the example.
So on this slide, it means that different splicing alternatives.
Yes, yes. Now the reason behind is actually you can see over here. That in exon 5, then they can bind to the lipid. Okay, so this is particular, for example, in the red cell. So red cell contain the enzyme as well, because then they can, circulate around and then metabolize any acetylcholine that is released and then diffused out from the terminals. Okay. Now you can see that this is exon 4. This one is the one that with the cysteine residues. Okay, so on. Now there can be homodimer. One of you asked about in the neuromuscular junction. You can see that actually there can be multimer all together, so that can metabolize the acetylcholine at the neuromuscular junction very quick. So this is very important. Now this is also happens in the brain as well. Okay, in the membrane lipid. So to metabolize the acetylcholine as quick as possible. Okay. So those are the different forms that can link together for different structures. You can see that you want some of those are free, around the tissues, and then some of those are in the red blood cell. Some of those are linked in the neuromuscular junctions, so they have a high concentration, so that they can metabolize acetylcholine as fast as possible. And that's the reason why that they can. The signal transductions can be just 1 millisecond, and then that's it. So let's look at the mechanism of how the enzyme metabolize the acetylcholine actually. So actually, the structure and the mechanism is very simple. Now, the initial step is actually by the ionic. So there's a pocket in the enzyme that can attract the acetylcholine into the pocket. And then that's the catalytic pocket. Now there's a anion site that can, attract the choline group. And there's a pocket that actually can bind, can attract the acyl group, and this is the asteroid site, in which that here is what happens, what hydrolysis can happen, so both by the ionic bond between here and the van der Waals force of the methyl group over here. So then sit into the pocket. So first, the second step, so first step is actually the acetylcholine attract to the pocket by the anionic and the van der Waals force. This is a methyl group, and then this is the anionic attractions. So once that is in the pocket, then it's ready for the enzyme oscillations. What happens is that you can see that at the asteroid site, the enzyme will go through the oscillations. Okay, this in the serine will donate the the proton to the choline, and then the oscillations of the enzyme over here with the serine. Okay. So you see that the hydrogen atoms is donated to the choline. Now, then the choline will release. It's okay? Very simple. Now, another thing that I want to tell you that I will not test you for the structure. Okay, that is not, so mostly doing for the exam is more of the concept. You know, no structure, because that is not needed for you. Okay, so I will not ask you to draw this out. Okay, those are the things that I will not test. Okay, because this is not relevant for your assessment. So, but we have to talk about it because then you will know that how the actually inhibitors will work. Okay. So then temporarily, then the oscillations of the enzyme, and then the choline will release it, and then that can be picked up and reused again. Now the other step is actually the last step is the oscillations. So this is actually the rate limiting stage. This is a transient enzyme with the water. So the hydrolysis of this RSI group by the water that actually helped by the imidazole group over here, the histine, okay, help to facilitate the proton acceptors and then help the RSI group to transfer it from the serine to the water. This is the last step. Now, this step is actually the time limiting step, and then actually a lot of drugs are acting on this step and then inhibit the enzyme for depending on the time scale. Some are fast, some are medium, some are long. Okay, it's okay. So very straightforward. So this is the equation. You can see that the summary. So enzyme with the serine donate the proton to the choline group. The choline is released, and then there's an oscillation of the enzyme, and then with the hydrolysis, with the help of the imidazole group from the histine, then that will release it. The RSI group, and that will become acetic acid. Okay. So inhibitions of the acetylcholinesterase. Now the inhibitors can bind to the active site of the enzyme and then prevent the hydrolysis of the acetylcholine. So if the enzyme is being blocked, then it cannot metabolize the acetylcholine. The acetylcholine will be there all the time. So that means what? It continues to excitations, open the receptors. That means that will be paralyzed the persons. And that means that actually you can, if you have drugs like that, that can kill the people. And which is really sad things that, you know, you see that that's the reason why that actually during the World War II or even World War I and II, that human synthesize, test all those different versions that can bind to this enzyme. So once it's bind, you continue that to activate it. So you may ask, so why it can kill people?
Because it's paralyzed those muscles for breathing.
Yes, yes. Mostly paralyze the respirations, and that will, you know, the person will die very quickly. Now, that's the reason, also the reason why that, you know, the Botox actually also acting on this pathway. It blocks the release, okay? So that means that it cannot move, same thing. So you can see that actually, it can still, the same mechanism, different mechanism, but same outcome. So one is blocking the release, then there's no actions. One is actually activating too much, then you cannot transmit either. Okay, so this is the idea about how you can act on different levels and then can still modify the transmission. Is it okay? Okay, we'll have more example of those. So, there are different drugs that actually can do this. There are three main types: the short-acting neostigmine, physostigmine. Now, another thing is that I will not ask you to memorize those names, those drug names. I will not test you on those either, because you are not the one that needs to remember those unless you are doing pharmacology or you are a doctor. If you need to know those. There are medium-acting neostigmine, physostigmine, irreversible, the important class of the toxin, organophosphate. So now this organophosphate insecticide, one is that actually can act on the insect. That's the reason if you have used the insecticide on cockroach, so and you spray the insecticide to the cockroach will then turn around and then moving really crazy. After a while, then they die. So same ideas actually. Now that's the reason why that across the organism they both use similar mechanism. So that's the reason why that some of those toxins can also act on us as well. So the effects of the anti-cholinesterase drugs are result in enhancing the transmission of the cholinergic transmissions, okay, particularly in the neuromuscular junction. Okay, now some of those can also cause the blood-brain barrier. Now those particularly organophosphate, so that will be a problem because that can cause and then have effects on the CNS as well. Now, the other effects, apart from the muscle, it can also cause the autonomic effects as well. Because if you still remember, both sympathetic and parasympathetic system also use the acetylcholine as a transmissions. Okay, the parasympathetic system, both the organ and the postganglionic both contain use the acetylcholine. In the sympathetic, that's a little bit different. That actually is the postganglionic, the preganglionic neurons only. Okay, so that's the reason why that it will affect the heart rate, bradycardia, hypotensions, bronchial constrictions, all those effects. Okay. Now, in the muscle, the neuromuscular junctions that certainly is the place now that will cause the fasciculations. It's just like cramps because of continuous action on those. So poisoning usually by exposure to the insecticide or nerve gas. As we'll talk a little bit more about those poisoning. That's the reason why that if a high level of poisoning actually is lethal actually, okay? And it needs to have the antidote within few minutes, otherwise the person will die actually. Now, but they can be also used as the drugs for some of the diseases, or use some for as the insecticides. That can be used as a classification as well. Now, therapeutic uses, Myasthenia gravis and glaucoma. We will talk a little bit about those two diseases, how the acetylcholine esterase inhibitors can be used to treat those conditions. Certainly, those conditions will be used some of the short or medium inhibitors, short time or medium time inhibitors. Now, the insecticide, parathion and malathion, those can be used. And chemical toxins, you may have heard about the sarin or soman. Those are the ones that actually used a lot in the or VX as well, in which that actually a lot of countries actually synthesize a lot of those and store those, okay, until well, nowadays still, countries will store those toxins actually. That actually is enough for killing all the people on Earth actually, at one point, okay? Particularly US actually synthesize a lot of those actually. By modes of actions, so you can also classify by the mode of actions, reversible or irreversible, okay? So clinical use of the anticholinesterase anesthesia, that, for example, used in the actions of non-depolarizing neuromuscular blocking drugs. We will talk about this later, how that during the surgery, people need the doctors need to block the muscle from moving. So you may ask why? Because the anesthesia actually have two steps. One is to stop your pain or your consciousness. Another one is stop your muscle from moving. So as you can see that, even during your sleep, actually, your brain actually send out a signal that actually for your muscle not to able to move. That's the reason why, otherwise, if you are still sleeping and you are moving, that's not a good thing, okay? In the surgery, same as well. So one drug will make you lose consciousness so that you're not able to feel the pain. Another one will be stop your muscle from moving. Okay, so that's the reason why that, in some of the cases, I don't know whether you heard about it, the patients going through surgery, he or she cannot move, but can still feel or smell. There are cases like that, actually. So that's the reason why being an anesthesia doctor will be very important. Okay, what is the, so this is a muscarinic acetylcholine receptors, okay? So if this is N, then that will be nicotinic, all right? So some of those that just not need to, have the whole name bring out, okay? So in the case of glaucoma, there's another ecophypate as a side drops. So this one needs to be localized, otherwise that will go to the other places, and that will affect the other functions. Okay, the treatment of myasthenia gravis, or in the case of Alzheimer's disease, we'll talk about this later. The Alzheimer's disease actually we'll talk about in the time that we talk about the disease at the almost at the end of the course. So we can see that again, there's a long-term, medium, and short-term that can be used in different places for different treatment. Here is just a table for your information. Okay, so some of short-term, medium-term, and long-term. The long-term one are mostly actually for killing or in the insecticide. Okay. So what is the actions of the drugs actually? So in which the drugs actually, you can see that this is a reversible, or this is irreversible. What happens is that the drugs will come into the pocket, okay, similar, and then form the structure, okay. Now this is the carbamide transfer, and then this one is the organophosphate transfer. So this group binds to the serine. Now the problem of it is that once it's bind, so metabolize. So what you need to do is actually have a structure compound similar that can bind, that can sit into the structure and transfer a group to the serine. And then if this group requires more time to release, then there will be a problem already, okay? And then in the case of the organophosphate, same thing. This one is diphosphate, transferred it, the group to it, and then that will be there, and this will be, if this is not released, then that will be permanent, okay, this one. So now it depends on how fast this group release out. So that's the reason why there's a long-term, short time, long time, medium time, and then irreversible. Now here you will see, ask why is it reversible. This is the antidote that can help to reactivate the enzyme. Now if it's not reactivations, then the enzyme after a long time they will be dead. Okay, so you may ask why if the enzyme is inhibited, why it cannot produce more. So you see that the enzyme cannot produce that at a short time. So that's the reason why that once you inhibit it, then that will cause the problem. Okay. So this is just to show that there's a pocket actually that fit the acetylcholine. Okay, they contain the serine. This is the active site, the serine group, and then there's a pocket that that's the reason why you can design drugs that sit in the pocket and then inhibit it, the function. Now the organophosphorus and anticholine esterase, there are many groups that can form. So after the long time study, investigations, then you can design something like this, in which the R can be aromatic groups and or hydrogen X can be anything hydrolyzed, and this is just for your information. Now, at once you can see that if you decide something is more hydrophobic, then that can cause the blood-brain barrier. Okay. Now, if you develop some is less able, more ionic, then that cannot cause through the blood-brain barrier. So that will be more localized. As we mentioned that in the insecticide, there are parathion and the malathion, depends on those. So this one's actually pretty toxic. That's not a very good one. The malathion is better because that is safer in is more metabolized in human, so that is safer in human. Okay, some of the example of those. So here, this one is the chemical toxin, the wall gas. So you can see that this is sarin. Sarin is actually, you might heard, well, maybe you haven't. Yes. How do you know? You still remember?
Because the guy get executed a few years ago, and there's.
I guess you guys are not even born yet, or even.
Because I like animes. That's why I know.
You like what? Oh, animes. Oh, I see. That's the reason. Yeah. That time is the sarin actually. So this is the gas. That time the guy, I don't know why he released those in the subway. Did they find out what's the reason?
He don't like the world. Yeah, he said purified world. Yeah, just like that.
Purified well. He was a. The online student also know about this, crazy religion. I think the cow. Yes, right. The cow. Yeah, the religion. Well, religion or cow. That one is the cow definitely. Whenever it's depending on one human, then that usually is not something right. So you guys are pretty good, huh? I thought that you guys don't know about this because of Japan. Okay, I see. Yes. So that's the sarin. There's soman and then tarpon as well. So they are nerve gas, and you can see that they are designed to be very lipid soluble. You see that there's no ionic group. See that how how hydrophobic it is. So you see, they designed it actually very hydrophobic so that it can cross through the wrapping barrier. Now that's a double hit because that apart from the peripheral autonomic or muscle, that can also affect your brain as well. So that's the reason why all those symptoms, so sweating, dimmed visions, those contain the autonomic nervous system, vomiting, aphasia, convulsion, and paralysis. So you can see that the patient will die in 2 to 10 minutes. I think the silent attack actually kills some people, not many, I think. 20 something. Wow. See, that's the really potent drugs that can kill people. So there are possible ways to remove it. You know, you can remove the phospho group, but it is extremely slow. Okay, for example, this is the case. Once it's attached, then it will takes a long time to release it. So let's look at it. Now, but once it's binding for a longer time, then that will be irreversible. Actually, it's called aging. Once that the phospho group actually go through further metabolism, remove of the R, and then that bond will be even more stronger. And at that time, then even if you have the antidote, that cannot be reversed. Okay. Now, for example, the enzyme can be reactivated by, in this case, the hydroxyamine. Hydroxyamine, for example, if there is a hydroxyamine come to attract the group, and then that can release the phosphate group, and then that can reactivate the enzyme again. So those are the antidote that how that work. So again, you don't need to worry about those structure. Okay. Now, some of the effects are parsidomine, PAM, and if that actually plus the PAM, then that can actually result in the 100% survival. So those are the drugs that can save the people, but obviously that needs to be done in a really short time. Okay. Sorry. So this is the last slide. Now, this is a recovery of the acetylcholinesterase synthesis of the enzyme, but that will take a long time because from the transcription, translation to the production, that will take a long time. Okay. The way is actually to have the antidote. For example, the oxim similar antidote plus atropine. Atropine is actually the muscarinic acetylcholine receptor antagonist. So that actually protect the heart, okay? Because the acetylcholinesterase will stop the heart from beating. You know, the parasympathetic system will slow down the heart beating. Okay, so that's the reason why that it is can be used as the antidote. Okay, this one is particularly important. For example, if you heard about from the news and from when the time that is also a long time ago, the Gulf War, the first Gulf War or the second Gulf War, all those U.S. soldiers going to Iraq, they carry the protective gear, and also they have the antidote as well. This is the antidote that they have, the oxim and the atropine. Now, oxim cannot go through the CNS. The atropine actually is protecting the heart mainly, and also the peripheral nervous system and the central nervous system from overactivating. Okay. Okay, so we will stop here today. Any questions? No, very straightforward. Okay. So the online student, any questions?
Actually, on page 27.
I will answer your exam questions later. Page 27?
27. As antidote. So if you don't have the antidote, that thing will just stick there, and therefore it's irreversible.
Yes.
Okay.
Yes. So when will the midterm be decided? Well, the midterm is online, and I'm making the questions at the moment. Yeah, should be true false matching and multiple choice. Huh?
You need to memorize spelling.
No. Well, you still have to recognize them. Yes. Yeah. So I don't know whether it's lucky or not lucky. So it's online. We have to do it online. Okay. Okay. Any other questions? I need to whether it's open or not. I'll tell you on Monday, next Monday. I'll tell you whether it's open I'll tell you, but I guess most of you like open, huh? Oh, okay. Oh, okay. Yes. Well, the reason for open is like that. Yes, I know.
You can't trust anyone to be honest if closed book.
Yes, we all know. Yeah, that's the problem. Yeah, we don't like it that way. Yes, but okay, I will decide it on Monday. But most likely is multiple choice, matching, and true false. So that shouldn't be a problem. Okay. Okay, if no more questions, then we'll end here, and then I'll see you next week.